I know that in undergrad you got a job washing fly bottles on the weekends, and
Yeah,
you said this sort of lab environment is something I could do. And
yeah.
then later you went into graduate school at Yale initially. I'm wondering, I didn't hear much about, graduate school to me seems like a large step, like a commitment into an academic career field. Were you intimidated? Were you confident that you wanted to do lab science for the rest of your life?
I I wasn't sure what I wanted to do for the rest of my life. Um, I think because I liked being in the lab, because I liked doing science, it it seemed like, well, do that now. Go to graduate school. It's I knew that I to didn't want to be a didn't want to go into medicine. Didn't It just seemed like what I wanted to do. Go into the lab, discover stuff.
Right. So it was kind of just a pursuit of passions. These are the things that interested you and
Yeah,
a
right.
logical way forward.
It wasn't, yeah, I can't say it was practical in the sense of thinking about a job in where it was going to happen to me and then in 10 years down the road or something like that.
And then I'm also curious about in 1981, your lab, or you moved back to Princeton with your lab. Um,
Yeah.
what caused this move back to the United States, if there was?
Ah, okay. So it was obviously it was that I ended up in Europe was an accident.
Right.
I had never been out of the United States, and eventually the man that I had, Walter Gehring, whose lab I was working in New Haven, had an offer to go back to to Switzerland, and he decided to go, and he offered me that I could go with him. I was actually the only person in his lab that went over to Basel, and it was easy for me. I was kind of young and unattached, and like I said, I'd never really been out of the United States, so it was like big adventure. And I lived. Then I did postdoc in Zurich, and then my first job was at the EMBL in Heidelberg, and it was about 10 years in Europe, and I'd convinced myself that I'd become very sophisticated and very European and could live in Europe forever. I the the EMBL at the time was a place was supposed to provide opportunities for young scientists, but not long-term careers. And so I had to think about getting a job or doing something like that. And the academic situation in Europe is more hierarchical and more structured. I there the full professors had associate professors under them who had assistant professors under them, and the option of it seemed to me that from what I understood of this in the States, you came back if you had a job at the university, uh, you were expected to build a lab of the same size as any full professor in the department. If you look and you can think of the general distribution, the way it generally is that assistant professors and associate professors and full professors at American universities have little independent lab units, and they're all they probably on average about the same size. And I knew that was what I want. I wanted that level of independence. Now the EMBL lab was a great place to be, and if you're a group leader there, you had access to lots of resources, and you didn't teach, you didn't have any obligations other than running your lab. So that was an attractive thing, but it wasn't going to be a long-term solution. So I had to figure out how what to do, and so I looked at jobs a little bit in Europe, and there weren't that many, and it wasn't where I wanted to be anyway from a scientific standpoint. And so I went on the job market, had an offer came to Princeton. Have been in New Jersey ever since.
Right. It's been a while now.
Yeah,
I'm wondering if you can talk a little bit about the sort of evolution of your field and your research since you arrived at Princeton. So, in introductory biology courses, they teach about the screen and the
yeah.
sort of, that's the level we get into. I'm wondering what
Okay,
the evolution
yeah.
of has been
So it
since.
is an interesting idea, interesting time. Uh, the screen was a great experiment to have done, and people had never approached developmental biology that way before. Uh, but and so it would have been and was interesting immediately when we did it. What of course made it have the impact that it really had on the world was that while Yanni and I were doing the screen, other scientists all over the world were not really interested in the kind of stuff we were doing, but were struggling to get the practicalities of molecular biology working, cloning genes and
Hmm.
sequencing, all of those things. And so by the time we'd completed the screen and had characterized this, a terribly complicated process of development, but that you could see it in terms of parts and interactions and hierarchies and steps, and all of that described in terms of gene activity, it had become possible to clone genes, identify genes, analyze those genes, analyze what the proteins they make, figure out if those proteins were fly specific or whether they were found in all species and whether the pathways work the same. And that process, which is essentially what happened from say 1981 to for the next 10 years, the decade, the 80s was this extraordinarily transformative time in that molecular biology was suddenly possible. It was even possible if you had never done molecular biology to learn how to do it well enough to clone genes and understand all of those technologies, and having a biological problem that was as well defined as the early flight development, and the fact that the genes that were identified in flies had homologs in humans that had similar activities, was all of that was kind of worked out in the next 10 to 15 years. And that kind of transformed how science was done, how this central role of that you could understand things that were really complicated by using an entry, uh, a genetic kind of entry into this broad problem. The great power of genetics is that you don't have to know very much to do it. You just, kind of bash away, and then things happen, and then you kind of collect and see what you get and think and organize, and you learn stuff. And you learn stuff without the biases of our preconceptions that histone proteins are important, or that acetylcholinesterase. If you
Right.
up to that point, until you had the concept of the genetic approach, you'd always start as a scientist from your very limited knowledge and a limited bias, and then try to get tubulin. Very often you choose something because, well, it was abundant enough that you could do biochemistry on it or something. And so that was the transform. That's, I think, an enormous transformation that happened in the field. The transformation of development, and then therefore also, I think, cell biology into a more genetically, molecularly defined field. And that gave all the molecular tools and handles to approach these were essentially cell biological questions, but at a very a much more mechanistic level. And you could choose the mechanistic level. You could get down into individual proteins and protein shapes and interactions, but you could stay somewhat some slightly higher or different level of interactions. And so that I think for the next 10, 15 years, that kind of transformed my own personal transformations. When I came back to the States, I decided that I would continue obviously to do development, but that I saw development increasing as a cell biology problem.
Right.
So, can we understand the visual morphological changes that occur in an embryo? Can we understand it in terms of cell biology? And does the power of the embryo give us a tool to really understand cell biological processes in a way that you wouldn't if you just had tissue culture cells? So, I mean, so there's this push. And I think that's 10, 15 years, that is the story. The other great technology that was transformative and that I didn't appreciate as much was computer science.
Hmm.
Everybody saw that happening at some level, and it was kind of happening in the background, and but to the point where at some level, say around 1995, 2000, it attempted to transform the questions that we ask into this larger scale, what we call genomics questions, or the big data questions that because the molecular technologies had reached a point where you could do big data, and it would have been pointless if you didn't have a tool that would allow you to search that data or to organize it. And so computer science gave that and kind of transformed the field in ways that I'm not sure have been totally useful, because I think there's a danger that you stay on a very superficial population level of molecules and not ask deeply mechanistic questions if you are too tied into the approaches, the big data approaches in the computer science. And even my friends often think about that you don't think about questions until you have all the data. You collect all the data and then you see what questions emerge out of the data and out of the observations that are possible. And that's certainly a valid way of thinking about science, but I think our brains are not better than that, but they're able to approach questions and define phenomena even before you have the data. And having questions is before you do experiments, and direct in a small scale science, have a question, and then small scale science figure what are all the genes that are required zygotically in a fly embryo to develop a normal pattern. Is a question that starts, and then you figure how can you address that question. Or another one would be, you have a bunch of mutants. This is in a genetic perspective. They all produce the same phenotype. How do you organize them into a pathway? How do you think about, so that's that initial question-driven approach is always going to be, I think, or not always, but most often is going to be a smaller kind of science than big data genomics.
Right.
And it's and so that's the transformation that we're in right now. So my own work, by about the late 1990s, it seemed that you could become more and more detailed, or I don't know quite why this happened to me, but I started hanging around physicists. And the nice thing about that is that uh, I'm very comfortable not knowing stuff, and um, I you or I'm very comfortable about knowing some details, but not having feeling like I have to know all the details. And I can tell you this: physicists have that arrogance in spades. That that they don't feel they need to know stuff, or that or they don't need to know details. So the whole goal is to abstract as soon as possible
Right.
away from details and get to and get to some fundamental generalization. And so, but the other interesting aspect of the interface with physicists is the tools that become possible. So we began using optical traps and things on early embryos back in the right after they were like developed. You could, if you hung around these people, they would have different kinds of ways of looking at things or optically examining or measuring phenomena. A lot of it has to do with measurement. A lot of it has to do with math and dealing with data and significance. I mean, it was a learning thing for me. Biologists kind of like, you have a sample and you have uh, an average value, a mean, and then you also have a variance. And biologists generally hate the variance, and they wish they want the variance to be small enough that their means of difference that they claim they've discovered something. Um, Physicists actually believe that there's information in the variance, and that you can extract how processes work
Mm.
by understanding the noise, understanding the variability, understanding. And to do that, you have to have a certain valid, sophisticated kind of math. And those things that made hanging with physicists and joint in collaborating with physicists so interesting. Was from the standpoint of cell biology or cell shapes or mechanics of cells or how they move or how a ball of cells would build a fold is essentially a physics and mechanics question. But then, things also like how an enhancer for a transcription, an enhancer would bind transcription or measure concentrations, measure uh, a concentrations of a transcriptional activator in a concentration dependent way that can produce accuracies of 10%, things that we can't do with the microscopes, but cells are able to do that. And so you have that approach is something that's really, really powerful and lets, since I know how to do stuff in the lab, it kind of allows me to bring something to an effort that I find very fascinating. Uh, and, a great opportunity to hang around these people who are so smart.